have time um, answer the questions. Finally, I'd like to thank all the three speakers um, um, and their dedication, because one person is joining quite early in the morning, another person is joining quite late in the night, and that's always a challenge for these global audiences. So without delay in this one, I would like to introduce my first speaker of this webinar. The first speaker is Dr. Kevin Eliceiri. He is a Walter Helmrich Professor of Medical Physics and Biomedical Engineering, and he is the Director of Center for the Quantitative Cell Imaging at the University of Wisconsin-Madison. His research focus involves developing novel optical imaging methods for investigating cellular microenvironment changes in wound healing and the development of software for multi-dimensional image analysis. His lab has contributed towards several open source imaging software packages, including FIJI, ImageJ, and Micro-Manager. In fact, the ImageJ was named by Nature as one of the top 10 computer codes that changed the science. And in 2022, Dr. Kevin was named an open hardware trailblazer by Open Source Hardware Association and Sloan Foundation. His talk will be on open source computational imaging of cellular microenvironment. And welcome, Kevin.
Thank you so much for the invite and for organizing this. I'm really excited to hear the other talks as well. I'm a professor of medical physics here at the University of Wisconsin-Madison and the Morgan Institute for Research. And today's talk is really about the cell side of medical physics. So I represent the community of medical physics is trying to bridge the cell to tissue gap, and very passionate about open hardware and particularly open optics and open software because the opportunity to link to medical imaging modalities and make them available. Hopefully some of the lessons we learn would also apply to other modalities as well, not just optics. But for a long time, my passion has been the cell microenvironment. And particularly, you see the picture here. If you look at the image here, the vast majority of this is intrinsic imaging. Your body glows. I'm fascinated by how we can extract the red NADH of the tumor, correlate the green macrophages and the white collagen, and how can we actually find a more complete picture of what's going on in the microenvironment of cancer and other wound healing phenomena. And to do that, we're trying to develop open tools. So just a disclosure, I am co-founder of Onlu, a medical device company that develops fluorescent sky surgery, and also consult with two companies that are involved in imaging and tumors. None of this work is in today's presentation, but I do think for the Q&A, it is interesting, when you make the decision whether to go open or go commercial, and I'd be happy to discuss that. So I'm an open source tool builder. I purposely put in some slides to kind of give context of our community. For those of you who are new, I'm very interested in building computational optics, which is a combination of software and hardware. And as I'll talk about a little later in the talk, very interested in smart imaging, the idea that you can bring in algorithms as part of the hardware, and the hardware knows where to look and help you in the imaging process. We're very interested in intrinsic fluorescence imaging. So if you know about fluorescence, it might be a newer concept that we're not adding in extrinsic fluorophores. We're looking at the most part for things that naturally glow in your body and the medical relevance of that, and particularly in pathology, but also in various kinds of biomedical models. And we're very interested in the fingerprint of your fluorescence. It turns out there are properties that can measure pH, hydrophobic, and metabolic phenomenon that are based on how long the fluorescence lasts, the lifetime, and also the spectra. And increasingly, to be relevant to our work in multi-scale imaging, which I'll talk briefly about, particularly ultrasound, MR, and PET, we're developing techniques to go. Optics is very high resolution, but very small field of view. And we're developing techniques to go as big as possible and do giant fields of view of tissues. And then a newer phenomenon that is kind of owing to our CT friends is optics is very rapidly going multi-directional. I won't have time to talk about that, but trying to actually have the full tomography at high resolution. So having subcell resolution, but in beyond 3D, many rotations. And then multi-scale is a big part of my role in medical physics. We absolutely need organ and tissue views, of course, which is the stalwart of medical physics. But so many of the phenomenons of, like, for example, metastasis happen at the cell scale. So going from cell, even subcellular, to tissue scale is really important. And so for the vast majority of my career, I've been developing open source software. And I only will talk about this a little bit. I'm going to focus more on the hardware today, but tools that like ImageJ and FIJI, and also Micro-Manager and OpenScan, open source acquisition tools, which I will talk about briefly, and then increasingly machine learning, including smart microscopy, and making these accessible as we can. So, and then we've also have done quite a bit of work in trying to deal with file formats. Even though DICOM is very popular, there are many flavors of DICOM. In the field of optical imaging, there's over 180 other formats, believe it or not. And so we've done a lot of work with trying to develop tools to translate between all these. It's called bioformats. So the central theme of my work is community and resource building. We have a national center that is able to all of you. Our mission is to help you. This is focusing on optical imaging, but a lot of it also could be used for medical imaging. This is a P41 center with Broad Institute, where we're developing tools like ImageJ, and with a focus on cellular imaging, but also multimodal imaging. We also recently, in the last 4 years, have started Bioimaging North America. There was the recognition that there wasn't, unlike the rest of the world where you have global bioimaging and Euro bioimaging, that in the US there was a gap in expertise for imaging, particularly optics. And so this is a national network that we'd love to do more with APM and other organizations to share ideas and expertise. And this actually funds quite a bit of expertise training in the imaging realm, particularly in optics. We also have a Beckman Center for smart light sheets. So this is the optical tomography I was mentioning. This also is a resource for folks that want to get 3D views at the cellular scale. We're also trying to move into the live human. So this is work trying to bring optics to the eye. And this is a lab at Wisconsin that is actually doing open source adaptive optics imaging of the retina. And then I think very important to this crowd is we realized there wasn't a good place to get help when you were doing image analysis. One of the biggest frontiers and challenges in all imaging, including medical and cellular imaging, is the image analysis. And so we have set up an ImageSC forum for all things open source and getting help with image analysis. And we have many, many partners, including representing all the major software partners. And this is a great place to get help for all things image analysis. And so if you type in ImageSC, you'll find that in Google. And then more recently, we formed a new center to look at multi-parametric imaging of tumor microenvironments, which is really what today's talk is about. And this center is trying to bring these tools I'm going to talk about in a few minutes to the overall community. So the focus of my lab is largely been on multiphoton methods, although we're increasingly moving into other methods as well, including ultrasound and other methods that could be bridges to medical imaging. We're very interested in those class techniques because of the fact that you can look at intrinsic cues, deep imaging, very good viability, and the vast opportunity for deep learning. For example, here on the right, you're seeing us use a combination of breast cancer of green fibers and red extrinsic labeling of a very important protein called syndicant involved in breast cancer. And how can we actually automatically find these signatures and help diagnose cancers like breast cancer? We've done quite a bit of work with multi-scale studies, trying to actually show how we can inform on other modalities, particularly ultrasound. There's great potential there for ultrasound and optics. And today's talk is really about cancers, but we have these approaches are very relevant, particularly the concept of the microenvironment, very relevant to many wound healing phenomena, including burns, diabetes. We've even done work recently in Alzheimer's, and very fascinated by this general idea of wound healing, starting with cancers, but going to many other types. And our interest in the microenvironment really is metabolism and ECM focused, particularly the collagen-rich ECM. And historically, we've done this through intervital imaging studies in putting human tumors into mice and zebrafish. But what you hear about today is just a real clinical opportunity to bring all this all to pathology. And there's great opportunity for pathology to be a bridge to medical imaging. A big problem in our field, though, overall in medical physics, is this image problem. And increasingly, we want, in all scales of medical imaging, whether it's optics or other modalities, we really want quantitative information. So before I dive into the hardware and the imaging, we have tried to develop a community here towards open source image informatics. So this workflow is really for optical imaging, but it would apply to other modalities as well. we've set up a complete open source paradigm for every single workflow. So whether you start with acquiring the data, the metadata, the quantitative analysis, data management, and I would argue this would be true of any workflow in medical imaging as well. And we developed tools for every step of the way. And so we developed analysis tools, we developed database tools, and I'll talk today, we developed some open source acquisition tools as well. And then of course the forum for answering questions. And so open source image informatics is a huge part of this community because you have to be able to process the data, understand it. So my talk summary is mostly going to be about our work in the tumor microenvironment, using these open tools, particularly looking at collagen. And this is really what motivates us. This is literally a needle in a haystack. This is actually collagen fibers stretching from a breast tumor, going to blood vessels that would go to the lung. And you can see this fluorescent cancer cell tightroping along to get there. And the very sad part about it is that anytime we saw this aligned fibers, the patient would die. This is a negative prognostic factor, has been proven to be very robust in many cancers, including pancreatic and breast. And we can actually track the cells in our rodent model and actually see these yellow cells go from the breast to the lung to prove they were metastatic. So we're very interested in trying to go after that. So we'll be covering that. Of course, we'll be covering a bit about open imaging in these markers. I won't have time too much to talk about the metabolism part, but it is a key part too. We're going to focus mostly on the collagen today. So now we're focusing on automating the analysis. How can we find these signatures automatically, the pathology workflows, deep learning of the trends, being as relevant as we can clinically, and making all these tools freely available. And then lastly, the opportunity for smart microscopy, which is really a big deal for pathologists. They want this information. They want to know what the alignment of the fibers are, where the placement of the cells are, the interaction between cells and fibrosis, but they don't want to analyze it. They want the answer given to them. And so we have to come up with ways where the microscope knows where to find these things and correlate to other imaging modalities and automatically give you an answer quantitatively. So open hardware is not easy. For most of my career, almost 25 years now, I've done software. And the software part is very well structured. There's very good tools for sharing and documenting software and a lot of best practices. But when you build open hardware, not just microscopes, when you get these complex systems like you see here that go beyond the commercial paradigm, there's really not a lot of good guidance. And home-built imaging systems, whether it's a microscope or something else, really drive the cutting edge. They usually lead the academic ventures, what lead then to the commercial ventures. So open source plays a role in facilitating industry as well. And we find that there's a real need to go beyond what's in the research journal. And there's very little, typically you get an image like this just showing an optical layout or a system diagram, but that doesn't help you build that system. And so we need to come up with ways of being transparent, reproducible. And part of the problem is that unlike software, there's no source code for instrumentation builds, not just optics, but any kind of instrument. There's no obvious CAD format for how do you build a system. And just knowing where they are is not enough. And in our field, we focused more on a single part rather than parts of parts. And in medical imaging of all types, it's very rare use a single part. It would be many parts together. And so how do you share and reuse that is a big challenge. So we've been working with the Open Source Hardware Association and also Sloan and many others of how to come up with a way of sharing and documenting your system, which I think could be true of any kind of hardware builds that are complex. And if you're interested in that, I'd be happy to talk and share. But in those open hardware efforts, we really are focusing on the multi-scale problem, cells and tissues. And many of you call most likely you're in this camp of really trying to understand MRI, CT, ultrasound for anatomical and metabolic. Most of my career has been here trying to look at cell resolution, but increasingly we're trying to show the relevance of this to medical imaging. And I, on purpose, drew this mesoscale gap. There is a gap that's not very well covered by any imaging modality. We're working hard with our collaborators to cover that as well. But for today, we're really interested in the submicron aspects. And that's what modern optical imaging can do for you, that you can actually look at the microenvironment and understand cells invading the matrix. You can look at non-invasive 3D, and you can even look at things that glow naturally, so not even extrinsic. And then what's really fascinating is that you can go after the chemistry and physics. And if you've worked before with optical imagers, you may think I mean the chemistry of the fluorophore or the physics of the microscope. There are certain reasons microscopes look like they do and the form factor. But for this talk, it's really more about the physics of the sample, which is really interesting from a medical imaging point of view. Things like stiffness and orientation are really important parts of the sample. A collagen orientation, which I'll talk about today, is a great example of that. And then I won't have time to talk about this too much, but we also can measure metabolism, and that's all about the chemistry. So I'm fascinated and driven by this image where you actually are looking at cells invading from the breast tumor, for example, metastasizing to, for example, the lung, and the interplay between macrophages and collagen fibers and other immune cell types in tumor cells, and trying to understand that. So we many years ago discovered a phenomenon where collagen realigns and the collagen comes in, and we thought it was actually macrophages secreting the collagen to stop the tumor, but over time we saw it become a metastatic highway. And we discovered this in breast cancer and pancreatic cancer, and now we've been up to about 20 other cancers where we see this phenomenon. But it's still limited in its impact in the field because the need for automating this and particularly bringing it to pathology. So we have been trying to develop a system that would automatically find this in a whole slide image. You could scan an entire patient sample and automatically find in breast cancer, for example, where the lesion is and where these collagen areas are, and trying to do it as much as possible, not requiring specific labels. Normally for these kind of studies, you would have a pathologist individually annotate everywhere, and it's just not practical. When you have a gigapixel image, was what you get when you have a large scan. You can't expect the pathologist to annotate that. It holds back the field. It's fine for research studies, but for clinical studies, they're not going to want to sit down and annotate. They feel if they have to do that, they might as well just do the study themselves. So we need to come up with a way of using weak labels or even unannotated labels. So we've been working a lot on this idea of weakly supervised learning, where you just have slide level labels, and you're basically just saying this image has a lesion, and that's enough to be positive. So this is called weakly supervised learning. And so the idea is that you really have images that are very weakly annotated, and it makes it much more practical. And so we took our system, made it open source. So this is the optics part of it. It's essentially a slide scanner that combines what you might know as a whole slide imager, like an Aperio, with the multiphoton that could do all the collagen imaging. And that part doesn't matter as much. It could be any kind of optical system. And then we integrated it with the machine learning and runtime processing so that we have a system that's quite complex, but all open source, where if you look at, you don't understand this fully, but we have open source acquisition system called Micro-Manager. We have a Python layer for calling all the deep learning, such as PyTorch. We have the GPU layer for processing that, and the various codes that control the microscope. And then you also have the option of a third-party open source package called QUPath, where we're able to annotate the image for the pathologist, giving them a friendly interface. So this system exists. It's been published open source. We actually have it installed in a collaborator's lab, a pathologist, and part of the system, and also in our lab. And the idea is to really make the system available to the community to allow you to do this workflow where you could bring in a whole slide image, and you could choose two workflows. If you want to automatically find the lesions in the collagen fibers, you can do that. In fact, you could even improve the image quality by denoising and so on, and then compare that automatically, our view with the classic H&E view. Or the pathologist could drive the acquisition without actually ever having to touch the hardware. They could draw in this very user-friendly package where the regions they want, and then the microscope will collect just where they tell it to. And we think there's a lot of potential there of this idea of a smart slide scanner. So in summary, collagen organization parameters are prognostic markers for cancer progression. We have shown that collagen fibers, so as you see here, are a huge part of cancer progression. We have introduced a whole bunch of various open source imaging and deep learning methods that can extract this from pathology samples. We have been focusing on pancreatic cancer, but also breast. Fibrosis is one of the major factors, and is negative prognostic. More aligned collagen means the patient unfortunately dies. And we've been trying to do deep learning on that and sharing these methods. We have many directions. One of the things that pathologists have asked us with these open tools is, can we tell apart hard things like chronic pancreatitis? For BDC, it's often misdiagnosed. So we're working on that more, also trying to automatically predict and find pancreatic cancer. We're also trying to work on ways of being able to find collagen data from pathology slides and ways of speeding this up. So we also have another grant to try to find ways, including FPGAs, that might have some advantage over GPUs for hardware acceleration. We are trying to implement at Cleveland Metro in Cleveland with our collaborator Agnes Loeffler, a runtime system where we have upgraded her microscope with some of this work where her pathology microscope is capable of seeing collagen fibers, see how it works in her workflows. We've also been trying to develop better tools to segment all the cells in the whole slide image and relate it to fibers. Huge potential, we think, for our metabolism work, where you can look at glycolysis and the Warburg effect as it relates to collagen. And then, like many groups, we're very interested in trying to bring in the spatial transomics field. Could we bring in genomic information that's present in a tissue and correlate that with the imaging? So with that, I think I'm almost out of time, but I'd like to thank all our funding from the Sloan and Oshma Foundation, NIH and CZI as well. Actually, I'm at a meeting right now that's CZI that is promoting open imaging, including medical imaging. And then many people in my lab, in particular, Mark Tashida, who did the bulk of the work that I talked about. And thanks to all of you for listening.
Thank you, Kevin. That was a very nice whirlwind of description of the tools. And we will have the question at the end. But now I want to welcome Dr. Heather Whitney. She is a research assistant professor in the Department of Radiology at the University of Chicago. She conducts research in CAD of breast and ovarian cancer, focusing on the modalities of dynamic contrast-enhanced MRI and ultrasound. Currently, her primary area of interest are in artificial intelligence and radiomics across the imaging and classification pipeline, from medical imaging acquisition to perform evaluation and data harmonization. She does research and collaborates in MIDRIC. It's called the Medical Imaging and Data Resource Center. Within MIDRIC, she works on methods of task-based distribution, interoperability between the data enclaves, and monitoring and studying the diversity and representativeness of the MIDRIC data common to foster research in AI and health disparity. Before she starts, I just want to let you all know there will be a CME or CPD will be available for all the participants who attend incomplete. The second is there is this couple of people have requested that not to do any chatting because that can be a disturbing for the audience. All your question, please put it on the Q&A because if you put it on the chat, it will not be followed. And I request all of you to refrain from chatting. And the title of the talk for Dr. Heather is Open Source Tools from MIDRIC. Here you go. Thank you.
Thank you, Mahesh. So thank you, everybody, for attending. I'm really pleased to share some open source tools that we have from MIDRIC. I am at the University of Chicago, and within MIDRIC, I'm a member of 4 groups that we have. We have a lot of things going on, a lot of things to share with you, so I'm excited about that. So just a quick overview, I'll be talking about MIDRIC and then data, tools, and resources. And the last point I think is very important. We're very interested in collaborating with institutions from all over the world to collect more data. So I hope that you'll consider that at the very end. So first, I want to share with you the data that's available for MIDRIC and in particular, the steps that are taken to make sure it's a very high quality. So MIDRIC, just a quick overview if you haven't heard about it already, it was established in 2020, and it was in response to the COVID pandemic, but is also moving now towards more generalizable applications. And it's an open and curated and diverse image data commons. It's a partnership between AAPM, American College of Radiology, and the Radiological Society of North America. And it's supported by the NIVIB through a contract hosted at UC and is on the Gen 3 data platform. We have currently over 24 institutions and over 100 investigators from all kinds of institutions. And there are 2 scientific components of MIDRIC. One is the Open Discovery Data Commons. As I'll discuss, we have already released over 148,000 imaging studies to the public, and that includes over 58,000 unique patients. And perhaps this is a really important element, the machine intelligence computational capabilities. This is not just an imaging database, but rather includes tools for accelerating AI research. And within MIDRIC, there are 5 technology development projects, of which I'm a member of 1, 4 data science committees, 4 working groups. I'm working within one of those, and 12 collaborative research projects. So we're also producing tools in addition to the data. And all of these are AI-ready imaging studies that are free to researchers. So I hope even at the end of this presentation, you might log in, set up an account, and start seeing what's available to you. So just a few comments about our data ingestion, curation, and release pipeline. We work with medical centers to intake the data, and we work with RSNA and ACR to collect these data with really high quality. And then it goes through curation, harmonization, annotation, all kinds of things to make it ready for you to work with. And this includes the contributions by AAPM medical physicists on these questions. And then AI and ML algorithm developers can work on things like cohort selection. They can download the images, do all kinds of things with those. And right now, our contributions are coming from 23 states. And as you'll see in a moment, we do have a few overseas contributors right now, and we are looking to have more. So a few comments about the curation and the harmonization of the images, which is, I think, very important because here is an example of a public COVID-19 DICOM chest X-ray image collection. And if you look very carefully, this is not a MIDRC data set, but rather a different publicly available one. And actually, this data set includes a knee or a set of knees in it, which is not otherwise annotated as such. So this is exactly the kind of problem that we are avoiding with the careful work done with MIDRC. And so we have multiple DICOM study descriptions from incoming MIDRC data. It's matched to a unique LOINC code, and this is derived from the NIBIB funded RADLEX. And this really increases our ability to make sure we've got high quality data. And this enables really efficient searching on our Gen3 user portal for cohort building. And you can see here, this is the overall pipeline of steps that takes place between when a data set is contributed and then when it's released to a user. So here's an example of this mapping from the DICOM metadata to the LOINC codes. There was previously a MIDRC seminar about this, and I encourage you to look into that further if you're interested. But what happens here is on the left, you see all the DICOM metadata that comes in, and as we know, there's some variability that happens within that. But then if you look over, for example, these two studies here, differences in the DICOM headers, through our LOINC mapping, this all gets harmonized, and it really facilitates a lot of imaging work. And so we have a multi-stage annotation process also behind the scenes. We employ natural language processing and local reports to extract key features. We curate annotations from external investigators where applicable. And then we'll have helper AI and a human expert to refine and verify these layers, these labels. And this is all going on in the context of our grand challenges, especially. I encourage you, if you want to just Google MIDRC grand challenges, you can see what's available there. We have one in process right now. And all of these data quality steps happen in the process of that. So right now at the MIDRC Data Commons, it's housed at data.midrc.org, and you can always see an updated figure of all the data we have available. I took this screenshot yesterday. So we're up to 58,000 unique cases that have been released to the public, and a number of different contributors and a number of different imaging modalities. You're welcome to look around there further. Here's a screenshot of what this the dashboard looks like if you're trying to collect a cohort, and you can select cases by the imaging study types. So for example, here is a screenshot of what it might look like if you were trying to select just chest X-ray imaging. You can also select by properties of cases, all kinds of different factors, but this is at data.midrc.org. And you can go from here, you can select your cohort. You can actually make it a repeatable cohort as well to ensure that other people can download that as well, and then move forward with some imaging studies that you're interested in. All of this is free and totally available to the public. We also have a data portal with a DICOM viewer, which makes it really easy as you're building your cohort, take a spot check of some images and get a sense of what you would be working with. We also do a lot of work on curating and monitoring the imaging to make sure it's representative of the population, or that a user can craft a cohort that is representative of the population they're working with. And I want to emphasize that we're actively seeking data contributions from rural and underrepresented systems, including internationally. And we believe that this is really important for the development of ethical and trustworthy machine learning algorithms. So here's an example of some data. I'm responsible for monitoring the representativeness and reporting that to the Bias and Diversity Working Group, where we discuss it. And so you can see here a quick snapshot of our most recent statistics on demographics. We have a paper that actually just came out yesterday on this, so I'll talk about that in just a moment. And of course, you can look at that paper further. And again, we are tracking the representativeness over time, because as different factors in the pandemic changed over time, and then as our contributors changed over time, and then moving forward, as different factors will influence the images we receive, these factors will change. And so, for example, this is our longitudinal monitoring of the combination of race and ethnicity over time here, again, referenced in our paper that was just released yesterday. We also have what I believe is a very unique opportunity within the data set for via the sequestration of the data that happens. So when data is ingested into MIDRC, we release about 80% of it to the public, and about 20% is completely sequestered. And this is completely restricted from public access, and this is for testing finalized algorithms, not development. And this is a really unique element because, remember, MIDRC's goal is all about facilitating and accelerating AI research and medical imaging, especially getting it to clinical translation. So via this sequestered data set, we're able to tell developers, you can test on this and we'll let you know what the results are. But you don't get to actually see the individual properties of those test cases. And this is also really desirable from the perspective of regulators such as FDA. So we do things such as in this process and check, just sure that users can know that the open data set does mirror the sequestered data set in these big picture ways. And we sample, and then we'll take the sequestered comments, and a user can come to us and say, okay, we want to test an algorithm, perhaps to distinguish between the cases with COVID and without COVID. And so our goal is to, within that sequestered data set, kind of do this task-based sampling procedure and offer our users the results of their algorithm, and then they can go back and they can revise their algorithm and then come back and test it again, but all within that protected commons, such that they never actually know the test set, and that helps with the regulatory process. So this is just kind of a quick overview of where our numbers are right now. We have ingested over 309,000 imaging studies at this point, and then released to the public. Our numbers are, I think, are even bumping up maybe in the last day or so to 162,000 imaging studies. And we have lots of registered users. We hope that you'll become one. And we have publications, again, 2 that just came out, one literally yesterday that we'll talk about, lots of presentations, and we're offering algorithms. So I'll talk about the GitHub offerings in just a moment. So different tools that are available to you through MIDRC. One is you can access what's called our bias awareness tree. And I understand these slides will be archived for your reference later on. You can get to any of these by Googling MIDRC and following the menus. But if you want to, there's a QR code here where you can get to this. And this is, again, this kind of extra element of MIDRC that we want to use to facilitate research. So in the Bias and Diversity Working Group, we're really interested in, across the whole AI pipeline, what are the sources of potential bias that can happen? And then how might users work on those, at the very least identify them, hopefully even mitigate them in their algorithmic work. And so if you think about the process of data collection, model development, model evaluation, and model deployment, all of these different, and data preparation as well. These are the types of biases that we've identified that could be present, and they might be happening in multiple elements of the pipeline. So we recently published a paper on this tool. And so, for example, you might identify which step in an AI pipeline you're working in. Let's say you really focus on data collection, then all of these biases are potentially possible within that process. But you can also note that some of them might exist also in others. So for example, here I have boxed off the ones with the blue circle. That's kind of the key for reading this. So exclusion bias, for example, happens in both data preparation and data collection. So you can click on our tool and review a catalog of possible biases to inform yourself. And here's the listing that will pop up in the tool. And then you can learn more about a possible bias, including how to measure and how to mitigate it. And then you can review other cross-references to other parts of the tree. So for example, let's say you're working in this area and you want to know more about temporal bias. Some information will pop up and it will inform you about what it is. It will talk about real world examples that you could be aware of, how it's measured and how it's mitigated. So you can use this tool kind of backwards and forwards. You can say, I'm working in this part of the pipeline. I want to understand what's possible. Or you can say, I think I'm seeing this and I want to work backwards and understand it more. So we also show here the references to other parts of the tree. We also have a new tool called a decision tree. And again, you can use this QR code if you want to grab that, or you can Google MIDRC and get to it through the menus. This is another new tool that we've developed. And the goal here is to walk users through, if you're going to sit down and do an AI ML task with medical imaging, how do you actually make decisions about your evaluation process? So you can take this tool and first identify the goal of your task. Let's say you're working on classification, then you would follow subsequent steps as you further refine your task goals. So for example, It asks you questions about what is your clinical classification task. Let's say you want to do a binary classification task. You need to consider the qualities of your reference standard. So if you identify that your reference standard has negligible variability, you proceed forward. And then there's other kind of things that will come up. And this one has a lot of options, but in the end, perhaps you can get to receiver operating characteristic analysis, learn more about that. And we put references and different resources to help you know that when you're using a performance metric, you've really followed the pathway of what's the rationale for that, what are the best practices, and what do you need to know. So again, all towards facilitating and accelerating AI ML development. So then I want to talk briefly about resources that are available to you. So we've talked about data, tools, and now resources. We have 2 recent publications that just came out in the JMI special section on global health, equity, bias, and diversity in AI and medical imaging. And here's one of the one that came out maybe about a month ago, and it describes the bias tree. And so the title here is Towards Fairness in Artificial Intelligence for Medical Image Analysis. And here are a few screenshots from the paper. You can see we put that big figure, that lovely iconic figure, also in table form to facilitate reading it. And then some tables with overviews. And this is available at JMI. It's open access. So again, if you just Google JMI, you can get to this paper very easily and read it. We also just yesterday published a paper measuring, reporting the longitudinal assessment of demographic representativeness in MIDRC. And this is also JMI open access. And so here you'll see our methods for measuring the longitudinal representativeness in the demographic categories of race, sex assigned at birth, ethnicity, et cetera. And then also our comparisons of these groups to the US Census, as well as the Centers for Disease Control and Prevention in terms of COVID positive counts. So here we also propose some methods to measure these using the Jensen-Shannon distance and reporting on what these values look like over time. And we will continue to do so, but this is our report as of about 2 years in. We also have other resources. We have a MIDRC YouTube channel where we record seminars, even just like this one, but we have our own set of seminars and you can watch all of those. They're really informative. You can get to the YouTube channel, but just by Googling MIDRC, it does have an underscore at the end of its username. And then we have a GitHub center where you can download different algorithms that are being developed by MIDRC investigators, information regarding the challenges, all kinds of really interesting things there. So I'd like to conclude by emphasizing that we would really love to see more data sharing, data donation to MIDRC, especially from users all over the world. And you can see here, we do actually have a few that have come in from other locations outside of outside North America. And we hope to have more. So if you are interested in this, donating your data to MIDRC can help with satisfying the new NIH DMS policy for scientific sharing. So again, all of these links you can get to by going to midric.org, but you can email the list, the support team if you're interested. And then here's a few list of items. This is taken right from the data contribution page. We have a YouTube video about data contributions, a reference document, how to prepare for things, potential funding opportunities to support data contribution, and you can contact us, of course. So a few acknowledgements in conclusion. Gen 3, MIDRC Central at UChicago, our wonderful administrators, as well as AAPM Program Manager Emily Townley. And of course, a big shout out to all of the groups that come together to make MIDRC happen and the funding as well. So I can take any questions at the end. You can also grab my contact information via email or the QR code here. I also have a QR code to these slides, and you can sign up for the MIDRC newsletter, of course, by Googling and just getting to midric.org and finding that there. Thank you all.
Thank you, Dr. Heather. It was really great information, and especially your point of stressing that we need diversified data for these AI tools, and especially if more people can donate from outside the US, that will really help the whole that training the tools. And this is really thank you. And of course, this information will be relayed again in the it will be it's recorded and it's going to be relayed for participants, attendees to sign on and call upon you. The next speaker is Dr. Robert Finnegan. Actually, it's very late in Australia. He's an Australian physicist, brings extensive experience of geophysics astrophysics, and medical physics. After he completed his PhD from the University of Sydney, he continued his work through research fellowships across multiple institutions, where he co-established the PlatPi library to facilitate accessible, high-quality tools for medical imaging and radiation therapy. Currently, Rob is also advancing his skills through clinical medical physics training at the Northern Sydney Cancer Centre. And it is with great pleasure I invite Dr. Robert Finnegan, and he's going to talk about unlocking medical imaging with PlatPi. Again, I'm going to reemphasize there is a CME and the CPD credits available for those who participate in the webinar or watch the webinar completely. Thank you. Go ahead, Robert.
Thank you very much for that kind introduction. It's a real privilege to be able to share with you a topic that I'm very passionate about, is open source software. I'm joining you today from Northern Sydney in New South Wales, Australia, and respectfully acknowledge the traditional owners of the land on which I live and work, the Camaragal people of the Eora Nation. I'd also like to take a moment to thank all of the people who have contributed to making this project what it is today, and in particular, give a huge thanks to Phil Clapp, who is a colleague, collaborator, and friend. He co-founded the PlatPi software library with me, and it certainly wouldn't be what it is today without him. So just to touch on a few of the benefits that I can see in open source software, I think it really helps to drive innovation by providing a practical way of exploring new ideas, rapidly disseminating new knowledge, and avoiding having to reinvent the wheel. The collaborative nature of open source software also lends itself well for people with different skills and different experiences to come together and contribute to a project. And it allows people to take code and adapt it in new and interesting ways. But last and certainly not least is that open source software is fun. It's enjoyable. You get to work with a whole range of people on some really interesting projects. PlatPi is a Python-based package, and we chose Python not only because we love it, but also because it's the most popular programming language in the world and has a very rich ecosystem for scientific analysis. It also continues a long tradition of open source software in science, where we really are standing on the shoulders of giants. So as I've explained, PlatPi is a Python package for image processing and analysis. And it sort of came about when I first started research and was really drawn in by the detailed and fascinating images that were being used to guide patient treatments. And after a little time in research, I had a bit of an epiphany that almost all of the data that we were using in radiotherapy could be represented as in an image-based format. So not only the patient imaging, but also things like the dose distribution, structures or contours as we call them, and a whole lot more, as you'll see. So I was motivated to develop a set of versatile tools that could be applied across a broad range of image processing tasks. We really think that PlatPi is for everyone. There's tools that would suit people who don't have much program experience or even very seasoned developers, and people from students to professors. There's a lot of ready-to-use tools that can simplify and automate tasks that you're probably doing quite often if you're in this space. And there's a lot of what are hopefully easy-to-use functions that you can plug into existing pipelines or use to develop new algorithms. And underlying a lot of what we do with PlatPi is a lot of functionality around advanced image visualization to move beyond just looking at patient imaging and start to dive deep into how algorithms are working under the hood. So to use PlatPi, you can install it using the Python package manager, pip, which you can just run like this, or you can download the source code from our GitHub, and there's a link at the end of this talk. To then use it, we provide a range of command line interface tools that you could run in your terminal, and I'll go through a few examples. If you've got some experience with Python, you can also just directly import specific PlatPi functions to use in your code. And for power users, PlatPi has some really nice functionality to operate in a server-client paradigm, where you can use this to use more powerful hardware for resource-intensive tasks, or even connect it to a DICOM system. So most clinical tools would come under this category. So I'm going to go through a range of features in Platypi that sort of span the typical things we would see when we have a new project. So let's just jump in. You can read more about it in our recently published paper, but the best thing to do would be to head over to the GitHub, where we have a whole lot of Google Colab notebooks. So you can actually try Platypi in the browser without having to download or install anything. We've already heard about different image formats, and certainly in radiotherapy, we have a whole range of research formats, or formats that are commonly used for research. And Platypi provides some really nice tools to convert. Kind of my favorite that I use all the time is the DICOM crawler, which, as the name suggests, searches down into an entire directory of DICOM files. It could have different patients, different modalities, and it'll sort, convert, and organize not just the imaging, but also the radiotherapy dose distributions and the structure sets into a nice form that you can then use for your projects. And I'd just like to mention another open source package in this space, which is being developed by the OSCAT team. And this is called PyDice, and is being developed to try to create research-ready data sets from a variety of DICOM sources. If you're anything like me, you probably find yourself writing the same 2 to 5 line functions all the time. And in Platypi, we really aim to reduce this boilerplate code and provide a lot of functions for simple things. So just as an example of that are some label operations, which are things that we might want to do to extract spatial information from contoured regions, like in this MRI of a head and neck showing the different glands outlined. So you can see we can simply extract out the centers of mass or bounding boxes. Creating image projections is a really useful way to visualize your data sets, and Platypi comes with a range of tools that can project your data along any given axis using a range of different functions. And shown here is a CT scan in the maximum intensity projection and the average intensity projection. This is quite flexible and powerful and can actually project along an arbitrary axis in 3D space. So I've just shown projections along a rotating angle here. This can be useful to visualize complex structures or to compare your projected image to what you might acquire at treatment, for example. Platypi can and has been integrated with other common packages that are used for feature extraction and image filtering. These include PyRadiomics and SimpleITK. And here's just a nice example of how we can visualize those different image filters applied to that same head and neck MRI scan. Next, on to image registration, which is a very common task of trying to optimize a transformation between 2 different medical images. These could be images of the same patient at a different time, or different patients, or different modalities even. The aim with Platypi was to try to create really simple, easy to use functionality. And with just one function, you can optimize a linear registration. So this includes things like your rigid and affine transforms. And we've also made it quite straightforward to play around with the different parameters you can use. This could help you derive an optimal set for your particular use case. And moving on from rigid registration, of course, is deformable registration. And in Platypi, we've got 2 methods implemented, which are the popular Demons-based and B-spline algorithms. And again, with just one line of code, you can get started, and then you can explore the parameter space quite simply to try to find what works best for your use case. Going beyond just calculating these transformations, Platypi comes with a range of tools to visualize and quantify different aspects of the deformable registration. So on the left, you can see the deformation vector field showing on a voxel level how the image was warped. We can visualize the magnitude to identify regions where we had to push or pull the image. And on the right, here's the Jacobian determinant, which is a mathematical operation that describes, or quantifies rather, the local expansion at any point and can be a rich source of information for research and clinically. Segmentation is a very common task, particularly in radiotherapy, and so there's a lot of research in medical physics devoted to this. And Platypi comes with some simple tools to perform basic auto-contouring of structures like the bone's lung and patient external contour, which are often used in subsequent steps in image processing pipelines. We also have a simple to use atlas-based segmentation approach. This is quite a popular algorithm, particularly in adaptive radiotherapy, where we use images of the same patient taken on different days to define auto-contours. And this is flexible and you can use it for different modalities and use cases. Of course, I wouldn't be able to discuss segmentation without deep learning, which in recent years has risen to the top to dominate image segmentation or auto-contouring tasks. And NNUNet is an absolutely fantastic open source package in this space. It's shown to provide very robust results across a wide number of cases. And while this is very easy to use in a standalone tool, we actually found it quite challenging to incorporate into some of our existing workflows. And so being open source, we were able to adapt this so that we can actually just run the NNUNet segmentation models with just one function. If you're anything like me, then this might be your thought process when trying out a new image segmentation technique. And while it might seem straightforward, it's not always simple to assess whether or not a particular segmentation tool is working. Commonly, we would compare it to some reference. So in this case, a contour drawn by a human. But again, if you're like me, you're probably wanting a few more numbers to go along with the pretty pictures. And for this, Platypi contains a lot of tools that can be used to generate similarity metrics. I won't go through these in detail, but we include metrics for volumetric assessment of similarity, including the popular surface dice metric, metrics based on the separation in physical space between delineations. And we've also included the clinically relevant added path length, and then statistical measures, which are quite common in machine learning to assess model performance. Even better than this is that if you have a set of contours, perhaps from two different algorithms or one algorithm and one manual observer, Platypi can generate automatic reports. And this just takes one function. It returns this nice figure, which you can save off as you go, and also a pandas data frame. You can then write out to a speech and analyze later. So the last type of functionality I'd like to describe in Platypi is that related to radiotherapy data, and in particular dose distributions. This is how we assess patient treatment plans. And Platypi provides some really nice tools to dive a bit deeper than the standard overlay you might have seen by using different color tables and other display tools. And here's an example that makes it easier to see regions of the dose distribution where there's a gradient, or the standard rainbow color map that makes it easy to identify high dose regions. Color maps for the low dose wash, which can be important to assess dose to healthy tissues, and can even combine it with projection data to get a nice overview of the 3D distribution. If we have delineations, then we can go one step further and compute things like the dose volume histogram, which is used widely in radiotherapy, and specific dose metrics as shown here. So if a picture is worth 1,000 words, then I would guess that an animation would at least be worth a few million. And Platypi has some great tools to turn the imaging data that you have, real patient data, into really nice graphics that can be effective tools to communicate to colleagues. And so we've got an example here of the projection data rotating round, a 4D scan from a patient with some heart contours, and then just a slice through the 3D volume. So all of these data are from the same patient, and just go to highlight how much richer effective visualization can be. So as well as these visualizations, actually every figure in this presentation was generated with Platypi. So just to wrap up, I'd like to go through one of the use cases and available tools in Platypi that I consider a crowning achievement of Platypi and also of open source software more generally. So to set the scene, radiotherapy for thoracic cancers benefit a large and growing number of patients, but in recent years, there's been increasing interest in the role of radiation dose to the heart, which can lead to increased risks of negative outcomes later in life, like heart disease. And while we have established some risk models, there's a lot of uncertainty in exactly how heart dose relates to these risks. And part of the problem is that the current clinical standard is to just treat the heart as a single structure. So we wanted to go one step further and start to look at the different cardiac structures which have different physiological functions and tissue types. And so automatic contouring was a solution to be able to look into those large retrospective data sets that we require to develop better risk models. I won't go through this in a lot of detail. You can read about it. It was published recently, but it uses 3 different modules, which we aim to improve the robustness of this tool. So I'm really proud to say that you can now run this automatic cardiac contouring tool on your own patient data using Platypi with just a single line of code. And we estimate that to date, around 25,000 CT scans have been delineated in this way from hospitals around the world. Like any research group, we have far more ideas than time, but there's a couple of really nice ideas that are on the horizon that we'd like to integrate into Platypi, but I won't go through them in detail today. So thank you very much for your attention. I hope I've convinced you to check out Platypi, and some details are there, and you can look into that more. Thank you.
Thank you very much, Robert. And I would like you to all of the speakers to turn on their camera and stop sharing. There are several questions here. It's really impressive how many people around the world are interested in this topic. We have right now almost 1,400 participants signed on to this webinar, attending that, and a lot more will be viewing your tools, your talk later, and I like to appreciate. And I see that already you, Kevin or Heather, have answered some of the questions. But let me ask you some of the few questions, because we do not have time for all the questions. I also want to express that many have expressed that your talks were excellent. So that also gives credit to your work, and I appreciate this one. Let me start with this question to Kevin. What he's talking, this one question is like, whether in the optical imaging, is it an invasive or non-invasive procedure? Further, is there anything to do with optical density here to view at the cellular level?
Yeah, so optical imaging is limited in depth. Typically, you get up to about 1 millimeter, which is not practical for many kinds of in situ human imaging. So for the most part, optical imaging has been used in the human for epithelial, skin, and an eye, or where you have some sort of other access through an endoscope. But the work I showed was all on animal models or biopsy tissues. And there you can be quite non-invasive. In the case of a zebrafish model, or which is very clear, you can see through it, no problem. You can put a window in the mouse. Or in the case of biopsy tissues, you can be label-free and non-invasive on the RD section tissue. And so it really depends on your definition of non-invasive. But certainly on ex vivo tissues that are grown like organoids or biopsies or animal models is not a problem. But an in situ human, you're limited in depth. As far as cell density, there's big interest in understanding what the cells do in relation to the fibers in the TME. And there's big interest in understanding when you look at a high confluent cell microenvironment, such as in cancer, what is the interplay and the players and the percentages of immune cells like T cells with normal and human. And one of the real critiques of imaging over the last 50 years is that we isolate cancer cells by themselves and look at them in dense and non-dense environments. That's not realistic. You need to have normal and tumors and immune cells. So the intersect and the density of tumor, normal, and immune is quite interesting. So.
Thank you. Heather, I have several questions for you, but I just want to focus on a few questions, what we can see. One thing, the question is, this is an interesting one. When we say the data is available in public domain, does this mean the use of these data sets and metadata in research no longer requires ethical review?
I would say the best way to answer that is to describe what has happened up until the point the data is released, and then to also emphasize that ethics can always be in play. I don't think we're ever kind of fully free and clear and can say we can never consider ethical questions. So in the data donation process, that's first handled at the institution level. And so there are several points an institution might take. For example, an IRB, for example, would take a look at what needs to be done to donate the data. So there's IRB coverage at the donation level. And then the MIDRC process includes a number of steps taken to ensure patient privacy. So under the ethics umbrella comes patient privacy. And so additional steps are taken in the data ingestion process that happens with Gen 3. So those are two elements of ethics concerns for medical imaging that are really thorough, happening through the MIDRC process. And then a user might have additional ethics concerns, such as, are they asking an ethical question in the first place? And that would be the responsibility of the user to make use of the data in an ethical way.
Thank you. Let me ask this question to Dr. Robert. I'm combining two questions. One is, what library did you use for DICOM RT? The next one is, do MATLAB systems still useful nowadays?
Sure. Okay. So for the first question, we use a lot of existing open source libraries for DICOM processing, including PyDICOM, SimpleITK, and RTUtils. The second question, is MATLAB still useful? I'll try to leave my personal opinion out of my answer, but I think there's space for every type of programming language and system that people find useful. And certainly a lot of physicists have used MATLAB for many years. So yes, definitely.
Kevin, a question for you is about what dynamic effects do you might find from photofluorescence decay?
Yes, I only hinted at it due to time, but it's fascinating that we all know that metabolism is a major driving factor of wound healing, particularly in cancer with the Warburg. And what's unique about optics is if you look at the fluorescence, how long it stays in the excited state, you can measure that. And it's not an absolute phenomenon. It's not just a fingerprint, it's a dynamic fingerprint that how long it stays in excited state is dependent upon the microenvironment. So it's been shown that you can differentiate free and bound NIH is one of the holy grails in cancer imaging, because this means you can detect callosis. So you can actually tell when you have more free NIH. So what that tells you is if you have Warburg. So we actually can map Warburg in a live tumor, which is quite unique. And so the ability to see cell scale and even subcellular changes in metabolism is a big advantage of looking at the decay of fluorescence. And so this is called fluorescence lifetime imaging, and quite powerful, quite complicated in math analysis, but is limited to animal models or accessible human tissues, not in situ in the human.
Thank you, Kevin. Heather, can I ask you a question? I'm trying to combine two questions together here. One is, how can you classify images by races? When we know today from genetics, there is no different human races, but only different ethnics. The second question is, what are some of the downside of the creation of this AI tool when the image data is limited to that particular country?
Yes, so we use kind of standard procedures in categorizing data. And specifically for race, this is a category that's considered self-reported race. And it's not meant to suggest any kind of biological construct. We know from a lot of different studies that race is more of a social construct, and it describes the experiences of individuals. And in fact, the Bias and Diversity Working Group is continuing to work more on how do categories of self-reported race, and not all individuals will report a self-reported race. They may say none, for example, and our data dictionary allows for that. But rather, we do know that these can correlate with social determinants of health. And so that's one reason why we don't advise kind of studying things by individual categories, for example. That would go under the category of ethical questions that should be asked with the data in the first place. But rather, for example, an individual might have kind of several factors that they self-report that could describe their experience. So for example, the neighborhood they live in, that might affect the type of healthcare they receive. And so all of these factors together, not just self-reported race, are of interest when researchers are looking into more demographic categories of what happens in medical imaging. Hope that's helpful. And then I post answer in the Q&A, but this question of how do AI ML algorithms relate to where is the data coming from in the first place? A best practice in algorithm development is to have a really carefully defined task in the first place. And part of that is being really specific about, are you designing a task for a lot of populations? You need to know if your data is truly coming from a lot of different populations, or are you doing algorithm development for a specific institution, for example? And so that careful definition can go either way. And so that's one reason why when we have people who want to develop generalizable algorithms, they absolutely need access to data from all over the world to achieve those goals. And so this is really essential for MIDRC and for our users. And then some may want to focus on even a particular neighborhood of patients going to one particular hospital. So all of that labeling can support all of those different goals.
Thank you. Robert, let me ask you a question. Again, compliments to your talk. And the question is like, are there any platform for training and learning some of the tools for research, preclinical and clinical use, especially for beginners? Second is, how much does it really on or duplicate the pi DICOM? And how do you integrate into a treatment planning system? Again, sorry, trying to combine a couple of questions in the same thing.
That's great. No, I appreciate it. So the best way to learn more about the functionality in Platypi would be to check out the examples page on the GitHub repository. If you just Google Python tutorial as well, if you want more general information. The second question about how much does Platypi rely on PyDICOM? The answer is a lot. We use PyDICOM to read in all the DICOM files, and we also extend it quite a lot by building a lot of tools to sort through DICOM directories, which PyDICOM doesn't natively have. In terms of integrating it into the TPS, that's I'd probably need a bit more time to answer that one, but the short answer would be by using DICOM communications with a server process set up using Platypi.
A question to Kevin. I think I'm also interested in this question. As a researcher, when do you say your research is going to be an open source tool versus when you're tempted to make it commercialized into a product?
Yeah, it's very problem specific, and I usually try to figure out what is the greatest benefit to the audience. You know, is a technology going to benefit the most from a commercial interaction, and will that be the best way of having clinical impact and getting into the hands of the most end users, versus technology that might be better equipped to be disseminated freely and openly and less of commercial interest. And it's a decision that once you dive into it isn't too difficult, and there's a lot of considerations, including the commercial landscape, getting advice from companies, clinicians, and also patent officials. For example, in our work of fluorescent scatter surgery, we realized that the complexity of that system and the clinical hurdles, including dealing with FDA approval, really made more sense to go commercial. But in some of our software tools that don't require FDA approval, it made more sense to be open source and be accessible. So it really is an application-driven decision. So,
Heather, a question for you. Again, I'm trying to combine two things. Do you have data for brachytherapy? And also, are these data related to cancer patient undergoing some type of a therapy like chemo or radiation? Are they just randomized data? And what is the population size? I think you might already mentioned it, but generally.
Yeah, so on population size, that question is best answered by, we have over 58,000 unique patients represented. Now, that doesn't speak to kind of how many CT images there are, for example. So you can check out data.midric.org, and that always has kind of a snapshot of that current breakdown by modality. And right now, our imaging has been all collected under kind of most immediately the questions of COVID. So while we have COVID positive and COVID negative patients, and some of those COVID negative patients include their images acquired for non-COVID related images, we don't at this time have any images specifically acquired maybe in the course of monitoring cancer treatment response. TCIA, the Cancer Imaging Archive, is of course a very important archive for that. And I encourage you to take a look there. But at this time, kind of separate things out that way. That being said, though, MIDRC is in the process of expanding to other modalities and other modalities, other disease questions. So look for more updates really soon on that question.
Robert, here is a question which was follow-up to your last question. And the question is, can we write some scripts and add this library to our TPS for great toolbars that we have?
If you can write those modules in Python, then it's definitely worth a shot. It's not something that I've done myself, but that's a great thing about open source software, that you're free to take it and use it however you like.
I think we are coming to the end of the time because we're already 13 minutes past the time. I think I will close here. And once again, I'd like to thank all of you for taking the time and preparing such excellent talk and staying within the time limit. That is really thank you. That allowed for us to share this. And for all the participants, I really thank you for making this worthwhile effort for IOMP to host these type of webinars. Please visit the iomp.org, and for any future webinar information will be there. Plus, there is also the recording will also be available within 24 hours. This will be there along with all the other webinars. Again, for many of you, there is one of our CME or CPD credits are also available. So, and I really thank you. And again, we're going to hopefully place along with the recording a folder of some of the tools spoken by the speakers here. We're going to kind of put it there for use, and you can also connect with the speaker if possible to ask for the question. And once again, this is Mahesh on behalf of the IOMP and also AAPM, because this is a joint webinar, and I'd like to thank both the organization for hosting this. And I also want to finally thank Dr. Magdalena Stova, who's behind the scene, who made this look so easy for me and for all of us without any hitch. And thank you. This completes the webinar for this month. Thank you.
Thank you.